Hello, my name is Christine Morales, and I'm going to teach you about the reproductive system, particularly how genes and hormones will determine gender. So firstly, sex chromosomes are what determine genetic sex. A genetic female will be consisted of two X chromosomes, one X chromosome from each parent, and a genetic male will be composed of both a X and a Y chromosome. The Y chromosome will come from dad, and the X chromosome will come from mom. This Y chromosome is actually necessary for the development of male reproductive organs, and therefore its presence or absence will determine whether development will continue along the male or the female lines. So the presence of a Y chromosome means that the individual will be male, even if they have multiple X chromosomes. So if they were an XXY. If the individual only inherits just a Y chromosome, nothing else, they will die because the bigger X chromosome is necessary for life, since it has a lot of the essential genes that the Y chromosome doesn't have. However, if an individual inherits just an X chromosome, they can live. They will be female. An example of this will be Turner syndrome. It's just that they won't have they won't be able to reproduce normally, so their lineage will die out. Once ovaries develop in the female fetus, one X chromosome in each cell of her body is inactivated and will condense into a clump of nuclear chromatin. This is known as a Barr body. Which chromosome is picked is completely random. The one from the maternal side may be active, and the paternal side will be inactive, or vice versa. But this inactivation occurs very, very early in development. So it actually happens before even cell division is completed, which means that all cells of a given tissue will have the same active X chromosome, whether it be from the mom side or from the dad side. So the reproductive structures will differentiate at the 7th week, and before differentiation they are known to be bipotential because you can't morphologically tell if the individual is male or female. This bipotential gonad will be made of two things, an outer cortex and an inner medulla. And if the appropriate signal is present, the medulla will develop into testes. If the signal is absent, instead the cortex will differentiate into ovarian tissue, and whichever one differentiates will also have the other part of the bipotential gonad sort of like regress. The bipotential internal genitalia are made of two pairs of accessory ducts, the Wolffian ducts and the Mullerian ducts. The Wolffian ducts are made from embryonic kidneys, and again, as development will proceed to be male or female, the other duct will degenerate. Things that compose the bipotential external genitalia include the genital tubercle, the urethral fold, the labioscrotal swelling, and the urethral groove. Sex determination also depends on the presence or absence of the SRY gene or the sex-determining region of the Y chromosome. If the SRY gene is present and active, the bipotential gonads will develop into testes. If the SRY gene is absent, then the gonads will instead develop into ovaries under the direction of female-specific genes. So here we have a karyotype, and you can see the much larger X chromosome and the very small Y chromosome. Very cute, very little. This is why the X chromosome is necessary for life. It's able to hold a lot more genes. And this image right here shows how combinations of different chromosomes will lead to a male or a female individual. This image shows on the left, this side, shows the development of the internal organs, whereas this image on the right shows the development of the external genitalia. So as mentioned before, if it is female, the cortex will form the ovary while the medulla regresses, and under certain hormones, the Mullerian duct will become the female parts, while the Wolffian duct will regress because a certain hormone is absent, which I will talk more about later. And if the individual is male, instead the cortex will regress, and the medulla is what will form the gonads, the testes. Whereas if the correct hormone is present, the Wolffian duct will form the internal organs of the male, whereas the Mullerian duct will regress. As far as external genitalia, if they are female, the genital tubercle becomes the clitoris, the urethral folds and grooves will form the labia minora and the opening of the vagina and the urethra, and the labioscrotal swellings are what form the labia majora. If the individual is male, the genital tubercle is what forms the glans penis, the urethral folds and grooves will form the shaft of the penis, and the labioscrotal swellings will form the shaft of the penis as well and the scrotum. So female and male external genitalia have their counterparts for the opposite gender. Okay, so now we're going to talk about male embryonic development and hormones. So again, the SRY gene is very important in determining whether or not an individual will become a male, and the SRY gene actually produces a protein that is known as the testes determining factor, or shorthand on this slide, TDF. TDF binds to DNA and will activate additional genes, and these protein products will then cause development of the gonal medulla into the testes. Testicular development doesn't require male sex hormones like testosterone, and that's because the developing embryo can't actually secrete testosterone until after the gonads differentiate into testes. But once the testes do differentiate, they will secrete the hormones, testosterone included, that will help influence development of the male and external genitalia. So these hormones are AMH, or the anti-Mullerian hormone, testosterone, and its derivative DHT, or dihydrotestosterone. The testicular Sertoli cells are what secrete glycoproteins and the AMH, whereas the interstitial cells, also known as Leydig cells, will secrete the androgens testosterone and DHT. Testosterone and DHT are the dominant steroid hormones in males, and both will bind to the same androgen receptor, but the two ligands will elicit different responses. So the role of AMH in male embryonic development and female embryonic development is that AMH will cause the Mullerian ducts to regress, and then the testosterone will change the Wolffian ducts into the male accessory structures like the epididymis, the vas deferens, and the seminal vesicle. Later in fetal development, fetal, not female, the testosterone will cause the migration of the testes from the abdomen into the scrotum, and the remaining male sex characteristics, like the external genitalia, will be controlled by DHT. So something that is important to note in male embryonic development is something called pseudohermaphroditism, and this is caused by the individual inheriting a defective gene for the 5-alpha reductase enzyme, which causes the conversion of testosterone to DHT. So although they are genetically male, they will have normal testosterone levels, but inadequate levels of DHT, which is what caused differentiation of the external genitalia. So things like the male external genitalia and the prostate gland won't develop fully during fetal development, so they come out of the womb looking like a baby girl. And they would be raised as a girl would raise generally because that's how the parents perceived them when they were born. But then they will reach like puberty age, and the testes will secrete testosterone, causing masculinization of the external genitalia, pubic hair growth, and a deepening voice. Now onto the female embryonic development. So an individual will become female if basically there are things missing that cause the individual to become male. So if they don't have the SRY gene, if there is no testicular AMH, if there is no testosterone, and if there is no DHT, the individual will become female. If there is no SRY gene, the cortex of the bipotential gonad will develop instead into ovarian tissue. If there's no testicular AMH, the hormone, the Mullerian ducts will develop into the upper portion of the vagina, the uterus, and the fallopian tubes. And if there is no testosterone, the Wolffian ducts will degenerate. And since testosterone's derivative is DHT, there will be no DHT, and therefore the external genitalia will instead take on female characteristics. And that is the end of my presentation on the determination of gender via genes and hormones.